let me see if we can get this going. There we go. First slide. So this is a little bit of a catchphrase that we tend to use. It's alive. And what's alive? Well, of course, the sample is alive. It's important to realize that once you take a sample from whatever living source you're taking it from, the sample and the cells in the sample very much remains alive. And if you look at the chain from sample collection and all the way to what happens with the sample, homogenization and analysis, we like to define 4 stages of sort of activity or stages of death. You have the first stage to the left here, where it's the sample collection, where in this case it's a patient sample. So here the patient is alive. If it's in the case of a say a small experimental animal, that stage usually ends by the death of the animal. And what we have then is that the removal of the sample, and the sample itself is very much still alive and experience a very traumatic event. You have a loss of blood flow, which leads to hypoxia and a switch to an anaerobic metabolism. And anaerobic metabolism is much less efficient at producing energy. So you have a low ATP, you start lactic acid fermentation, so you'll have lactic acid buildup. That will lead to lower pH, and so on and so on. So this cascades, the loss of the blood flow, really sets a whole cascade in motion. And these are quite powerful signals which the cell senses. And well, the cell doesn't know it's doomed, so it will try to sort of react to these signals. In the next phase, what we call the bioreactive termination, that's where you have a homogenate. So there's no longer sort of living cells. The cells have been broken up, the compartment have been broken up, so there's no sort of overall control. So it's more, but you still have active enzymes. And so that's a source of the change there. And as I said, the compartments are broken up, so you have a mix of substrate with new enzymes, which generally does not happen. And in the very final stages, we have chemical with oxidation and so on, non-enzymatic processes. We're looking at 2 of the stages here, and with the stabilizer system, and we want to remove the enzymatic processes that happen either in the first, sort of from the sampling to freezing, or during homogenization, which happens during sort of and after homogenization and extraction, which happens in the 3rd phase. The post-sampling changes are most easily seen if we look at peptides. And in this case, we see two panels. There are 2D representations of LC-MS runs, and we have retention time on the x-axis, and we have the molecular weight on the y-axis. The left image here has been inactivated, stabilized, using heat directly once it's been removed, and we see that the majority of the major, in this case spots here, have been identified as various neuropeptides. If we look on the right screen instead, the right panel, there we have an artificial 10-minute lag between the death of the animal. In this case, we're looking at mice brain, and between the death and the extraction and stabilization, we see loads and loads of spots. So there's lots of peptides in here. However, it's very difficult, on the verge of impossible, of identifying any of the sort of interesting neuropeptides that we know should be there. Instead, what we see here is a lot of degradation fragments from high abundant housekeeping proteins, and usually generally not very interesting. Phosphorylations, that's another protein phosphorylation, that's another molecule that will rapidly change post-sampling. In this case, we're again looking at mouse brain, and using a technique called reverse phase protein array. It's kind of a modernized way of dot plots. What we're looking at here as a postmortem time series, where we have 0 and then 10 and then 30 minutes postmortem time at room temperature, and we're following these 11 phosphorylations, and as you see, there's one group of them that very rapidly loses pretty much all phosphorylations. Some fare a little better, but nonetheless, it's a marked reduction compared to the initial levels. If we look at another study, we did the first phospho study. This study is by another group that we're not working with. I'm just including this to show the complexity of studying phosphorylations. The experimental setup is pretty much identical. They're looking at in this case, it's a post-excision. They're using human uterus tissue, and they're also looking relative to the level on the first time point here. In their case, they're seeing an initial increase for quite a few of the phosphorylations. Some of them decrease rapidly, and at some point here, 25 or so minutes post-excision, they see this rapid decrease. So not only are the post-sampling changes determined on the phosphorylation itself, it's most probably also depending on the tissue situation they're in. So studying phosphorylations is really tricky, and one needs to have control and to really know how they change post-sampling, unless you're stabilizing your sample and removing the enzymatic activity that causes these changes. A more recent example of another group of molecules is free fatty acids that also show a dramatic change post-sampling. In this case, we're looking at the Borén homogenate, and we're following these, I think it's 9 or so different free fatty acids, the release of these free fatty acids over time in this homogenate on a sort of minute scale up to 40 minutes. And we see that the increase for some is truly tremendous, with several 100% increase in the levels, where some are not so affected. But nonetheless, it's quite an increase. And so that's the problem that we're addressing, that in this tissue there are powerful sort of forces. There's enzyme activity which causes shifts to the analytes. And Denator have a solution that we work with thermal denaturation, and we have a system. It's called the Stabilizer T1. That's the instrument on the right. It's a benchtop instrument, and it plugs in the wall, and turn it on, and it's ready to run in roughly 5 minutes. It's pretty automated and pretty easy to run. It works with a sample card. We call them maintainer tissue, and that's where you place the sample card, and then it's closed and form a closed cavity here where the sample is during the treatment. There are also other cards, the maintainer liquid for more liquid samples, and there's also a maintainer DBS card for the dried blood spot application. So how does it work? What does it do? First of all, I think the next slide is sort of how is this integrated into the workflow. So in order for this to be efficient, it's important that it's implemented either very close to the source of the sample, so either next to the operating table if it's in a clinical setting, or next to wherever the experimental animal are situated and the samples are being extracted. And you sort of insert the sample into the maintained tissue and then directly into the stabilizer, and you have stabilization and inactivation of the enzymes roughly between 30 seconds and 60 seconds after you introduce the sample into the stabilizer. It's also possible to run this using frozen samples for a situation where it's difficult or impossible to get the stabilizer instrument close enough to where the close enough to where the source of the sample is. So then it's important to freeze the samples rapidly after sampling, and then the samples are introduced frozen into the stabilizer and stabilized. This is also good for biobank samples, or if you have samples that's sort of collected for multiple purpose uses, and you don't know ahead of time that they will be utilized to assess certain molecules which are good, which are preserved by stabilization. It could also be advantageous to stabilize them in the frozen form. That's generally the workflow. And so what do we do in the instrument? So the instrument works by heating the sample, and we see here a schematic of that. And what the heating does is that it changes the 3D structure of the proteins, of the enzymes. And that's because the proteins, the enzymes are very dependent on their 3D structure for activity. So what we do here is that we start out with the tissue, and this is in tissue, where the proteins then our enzymes are in an active natural state, and we add heat. We do that by contact heat, conductive contact heat, and as the sample heats up roughly 90 degrees centigrade, the enzymes will unfold into a non-active state, and then the samples are removed from the instrument and allowed to cool down, and we have hydrophobic collapse of the proteins. They form a new 3D structure, however, in a denatured state, which is a non-active state. And as this happens in the cell where you have a really high protein concentration, you will also sort of then end up in a sort of aggregate coagulate state together with lots of other proteins. Again, a non-active state. So this is what happens on a molecular level in the tissue or in the sample which is being stabilized. If you look at the most basic, what happens with enzyme activity in a sample that's been stabilized versus a sample which is not stabilized. Here we're looking at phosphatase activity, and we assess this by using a common phosphatase substrate. It's called PNPP, and when it's cleaved it turns yellow, and this yellow coloration is then measured at 405 nanometer. Again, this is mice brain extracted in one-time PBS, so we're expecting a lot of activity, and that what we see here in the left column here, the left bar, the one no inhibitors, really high levels of activity. If we add inhibitors, this is the phosphostop tablets from Roche cocktail. We see an expected drop, but nonetheless the levels are roughly 40–50% of the original level, so still quite a lot of residual activity also with the chemical inhibitors. If we do compare that to a sample which has been heat stabilized, which we see here in this orange bar, we see that level is really on the background level. If we look on the other side of the phosphorylation story, the kinase activity, we see a very similar story. Here we're using an array of peptides, and these peptides each contain a phosphotyrosine motif that can be phosphorylated by a phosphotyrosine. The sample is homogenized, again, I think it's in the PBS buffer, and incubated with this array. And if you do have kinase activity in that sample, it will specifically phosphorylate the substrates on the array. And then you're using a general PT100 tyrosine phosphorylation antibody to measure the phosphorylation level. And what we see here are the 18 most phosphorylated peptides on this array. And we see the gray bars are for the snap frozen samples. Some motifs are very highly phosphorylated, others not so. And if we compare that with the heat-stabilized, we see there's only 3 motifs that are a little bit phosphorylated. And if we add all this together, we see that there's an overall reduction of 99.6% of the kinase activity in the sample. As I mentioned earlier, the stabilizer is really an upstream event that happens very early in the chain of analysis here. So downstream of you can do all sorts of proteomics. That's where we're coming from, with lots of peptidomics and proteomics analysis, be it with Western blot or mass spectrometry, NMR, and so on. We're also so exactly peptides and phosphorylation. That will we see the main benefits. We're more and more looking into small molecules, and that's very broad. So you have lipids. We've seen that. We'll see that later on as well. Pharmaceuticals, ordinary natural metabolites like ATP as well are preserved. There's ample opportunity for an analysis downstream. That was sort of the basis of what the technology does and how it fits into actual workflows. In the following slides, I'm going to focus on a number of customer examples covering a range of analytes, the traditional peptides, protein modifications, small molecules, and I've also included a little section on RNA, because I generally get questions on that, as well as a new application that we think is quite interesting, pathogen inactivation by heat. Starting out with the customer examples on peptides, I have 3 examples. First, an ordinary peptidomics approach, LCMS based, and then there's a multi-imaging of a specific peptide, PEP19, and then there's an example of radioimmunoassay analysis of 3 peptides. And so, looking at the first peptide example, what we see here is the LC trace of peptides being extracted, so that low molecular weight peptides extracted from bovine hypothalamus. And the yellow or orange asterisks, they mark sort of known neuropeptides. In the upper panel, where we see the trace for the heat-stabilized sample, we can see that the majority or all of the major peaks have been attributed and named and identified as being sort of known neuropeptides. In the lower panel, which is a snap frozen sample, we see it's still possible to identify a few neuropeptides. However, the spectra is totally dominated by 2 peaks which are identified as Statmin fragments, the one marked in red asterisks. Statmin has previously been shown to be pretty much exclusively formed in a time-dependent manner post-sampling, specifically in brain sample. And it's a N-terminal peptide, 2 to 20 or 2 to 22 amino acids that are cleaved off from the protein Statmin and increase quite rapidly post-sampling. As you see in the upper panel in the heat state, it's still possible to detect them there, and that's mainly due to the fact that removing the hypothalamus from a cow skull takes roughly 30 minutes. So there's still a bit of post-mortem time on this sample, even so on. Yep. If we look at the number of neuropeptides, we see the heat-stabilized, we see 14 compared to 7, so twice the number. What's also interesting to note is that these 14 make up 52% of the identified peptides in this sample, whereas in the snap frozen, the 7 only make up 7. So there's a lot of more uninteresting high molecular weight housekeeping proteins, peptides, degradation peptides from those being identified that has to be sort of sifted through in order to find the neuropeptides. That was the first example, and this is published, and the majority of the examples that I have are published, and you can generally find a reference to the paper somewhere on the bottom of the slide. The next example uses imaging mass spectrometry. What we're looking at here is a thin section, a sagittal section of a mice brain, and it's been arrayed and then with a matrix, and then a MALDI has been used to image it in each pixel. And the left column, that's the snap frozen, and the right here is the stabilized sample. And here we're specifically looking at an M/Z value 6715, which has been identified as a neuropeptide called PEP19. And we see the distribution in the snap frozen and the stabilized roughly equivalent, slightly higher singular in the stabilized. What's interesting, however, is these two, the two lower panels, where we see lower masses, M/Z 1755 as well as 1393. These two have been identified as being fragments, shorter degraded versions of the PEP19 molecule. We see the distribution roughly the same, so that makes sense. But what's really interesting is that they're almost exclusively found in the snap frozen sample and not in the heat stabilized. And our interpretation, as well as the research that did this, Richard Goodwin and Per Andren, is that this is due to degradation during sample preparation. That during the time that from the sectioning, from the sampling to freezing to sectioning and so on, that these two fragments are formed during that process. The third example of peptides, we're looking at a small system of dynorphin, so dynorphin A and dynorphin B, which are broken down to this lev-enkephalin arginine. And what we're seeing here is that we look at the snap frozen sample, that's the black bars. We see rather, well, good levels, but then again, compared to the lev-enkephalin level, the parents have much lower level compared to the degradation fragment, or the fragment. If we instead compare the stabilized sample, here denoted by the orange bars with the D, we see that the parents are much higher in level compared to the degradation fragment that we see here. What's also interesting here is that these samples, they're extracted, they're rapidly frozen, and then the extraction starts by putting the sample into boiling 1 molar acetic acid for 5 minutes. So they're basically doing a heat inactivation of the enzymes, but that's apparently not sufficient. We still have this conversion into the degradation form in those samples. If we move on to proteins, I've included a couple of 2D gel samples. The first sample is mouse brain, and we have a standard 2D gel 4 to 7, PI 2 to 4 to 7, and the molecular weight covering the whole range from 16 to some 250 up here. And what we see here, the identified have been color-coded so that the red and the blue are in higher intensity in the snap frozen sample, whereas the green and this orange peach color is higher intensity in the stabilized samples. And what we see here is that we have a majority of the samples being upregulated, or higher intensity detected in the snap frozen samples being detected in the lower part of the gel compared to the ones that are detected in higher levels in the stabilized samples, which are generally sort of in the middle to higher part of the gel. Again, the general explanation for this is due to that these are formed due to degradation fragments. It's also, when we go into and look at these individually, they tend to be identified as proteins which should have occurred much higher on the gel. So the difference between the sort of detected molecular weight is generally large compared to the theoretical molecular weight in those that are higher intensity in the slab frozen samples. The other sample is actually yeast cells. These are yeast cells in suspension which has been heat stabilized. We generally do solid tissue samples, but we're more and more getting interested in doing various sort of liquid samples, plasma, blood, in this case suspended cells and so on. And we think that the general principle of enzyme activity generally applies to all types of samples. What we see here is the comparison between direct extraction and then what we see on the left, and that's in 8 molar urea with inhibitors. So a pretty hostile buffer to enzymes. And we specifically highlighted, it's seven low molecular weight, quite strong spots, which are prominently in the direct extracted compared to the that were stabilized first. Here they're pretty much non-existent in the stabilized sample. These 7 were identified as being shorter versions of enolase. And enolase is one of the proteins which is most often found in 2D gel experiments. And there's a hypothesis that this is due to this is very actively degraded and that it tends to change rapidly during the sample preparation process. Another example of 2D gels looking at whole proteins is from white blood cells, neutrophils. Here we have neutrophils. They're extracted or isolated from whole blood, and they're extremely rich in proteases. These are generally macrophages, is one type. That they generally sort of tend to, they're part. What they do is that they digest other stuff in the cell. So it's a very challenging sample where you have sort of this interesting mix of lots of interesting proteases, and of course also lots of them, when you homogenize it, a lot of vulnerable proteins. If we start from the right here, the C panel, we see the sample extracted using an ordinary 2 molar, or let's say 8 molar urea buffer with inhibitors. We see that there's just a few faint spots in the lower area here. They modified the buffer by adding the detergent MP40, markedly increased. There is still quite a lot of things in the low molecular weight, again indicating that there may be degradation going on. In the stabilizer, in the heat inactivated samples, we see a marked decrease in the number of spots in the lower area, as well as we get a more defined upper region where we have high molecular weight proteins, which this may be a gel artifact, but it may also be an effect of degradation. That was the examples on whole proteins. Now I move on to protein modification. First and primarily we're looking at phosphorylations here. The first example will be Western blot. And then there's a 2D gel using a procodiamond, that's a phosphor-specific stain. And then there's an MS approach, bottom-up phosphanalysis where you trypsinate and then use mass spectrometry in a shotgun fashion to identify your phosphorylated peptides. And I'm going to end up with a little Western blot looking at a simulation as well. The first example, we're looking here at an example, sort of the experimental setup was to look at stability after heat stabilization, keeping the sample at room temperature after it's been heat stabilized. Can we sort of still see that the levels that we see if we have to freeze it directly? The rationale for this is primarily in a sort of pathology setting where it's difficult to have, where the sampling and then the analysis generally takes between 30 minutes up to an hour in standard cases. We're looking here is the phosphorylation as the 202-204 phosphorylation, and the red bars indicate the snap frozen sample, and we see the difference between the sample that was frozen immediately and one that was 30 minutes on the lab bench prior to being frozen. We see a reduction of roughly 75%, which is quite expectable. If we do the same thing with a heat-stabilized sample, and we compare the sample that was stabilized immediately after excision and then frozen, compared to this stripe bar with yellow sides, where we have the sample that was heat-stabilized and then left on the lab bench for 30 minutes prior to being frozen. We see that the levels are really similar. And this is, as we saw earlier, with the phosphatase and the kinase activity, there basically no kinase, no phosphatase activity left, so the phosphorylations primarily stay the way they are. Another example is this PA-1, also similar levels after 30 minutes at room temperature after stabilization, whereas the non-stabilized pretty much again reduced temperature. In the article here, there's some 14 more examples of phosphorylation that behave basically the same. The second example of phosphorylated is phosphorylated proteins. This is in the 2D gel, and we're looking at a train of spots. The upper panels, the A and D, as the stabilized sample. The lower, the B and E panel, are the snap frozen sample. And the left two panels, A and B, is from this cis-trans isomerase proteins that were identified as 3 isoforms. It's identified at spot number 62, 66, and 79. And we see the different sort of intensity, the distribution between these 3 isoforms, and compare that to the distribution in the snap frozen sample. We identified in the same spots, but the distribution between the isoforms are completely different. And we see the same for this ATP synthase subunit alpha, where in the stabilized sample we see the sort of isoform 21 being the most dominant, whereas in the snap frozen sample the isoform detected in spot 40 is a much higher intensity. And the explanation for this is that we have a much higher phosphorylation grade. So the 62 have a higher phosphorylation compared to the 79 and the 40 versus the 21. So this is presumably an example of hyperphosphorylation that presumably happens in the extraction. This is a dilemma with doing just direct comparison. They look very different, but it's of course not possible to say, well, this is correct and this is not correct. But it begs the further investigation into what is the most in vivo-like representation of the isoforms of these two proteins. Another example of looking at phosphorylation, this time using affinity phospho enrichment followed by the MS analysis. So in this case we have a direct comparison again, heat-stabilized mice brain samples compared to snap frozen samples. The samples are extracted, 8 molar urea, then that's diluted to 2 molar urea, which is then diluted to 2 molar roughly, 1-2 molar, and then digested with trypsin. And these peptides are then affinity enriched. In this case, we've been using the phosphotyrosine specific antibody from cell signaling, and this experiment is actually done bi-cell signaling altogether. And then so you have a much enrichment of phosphotyrosine peptides. And what we see here is the distribution in the ratio, intensity ratio between the heat-stabilized samples compared to the snap-frozen samples. The majority, roughly 2/3, they show roughly similar distribution, similar intensities between the two sample types. Whereas what we see whereas roughly 1/3 show at least 50% higher intensity in the heat-stabilized samples, and there's some 60 or so peptides that can only be identified in the stabilized samples, whereas there's one that was only identified in the snap frozen. There's also a few which shows much lower levels in the heat stabilized, and this is most probably due to have the majority of action that happens post sampling is phosphatase activity, where you remove phosphorylation. There are, however, examples of also kinase-driven activity. The kinase activity is, of course, ATP dependent, so it will be more difficult for the kinases to be operable in a low ATP post-sampling environment, but that does happen. Let's see here. This is a sort of a narrowing down on the tau protein. By chance, there were 3 peptides from tau and from the tau protein, and what we see here is that 2 of the forms, one form, the middle one, was only detected in the stabilized samples. The other 2 are identified with significantly higher levels in the heat-stabilized samples, and it might not look very much the difference here, but then it's important to remember that the log2 scale on the axis there. The last example of the protein modification, I want to highlight a different type, Sumoylation. This is a small tag which is added to a protein tag, which is added to the proteins, generally in the degradation pathways. Sorry, let's see. Get that. Now, what we see here is that we're looking at a Western blot, and we're looking at using an antibody against the Sumo3, which is one of the versions of this sumo protein, and we're looking specifically at the trimer of this here. This is the same example that we saw earlier, looking at the phosphorylations, where the setup was 0 and 30 minutes on the lab bench. So F said that's the frozen samples, and ST are the stabilized samples. And we see here already at the 0-minute time point, we see a bit of difference between the snap frozen samples, the FSED samples, and the stabilized samples with slightly higher levels of the trimer in the frozen samples. If we, however, wait for the 30 minutes to pass, we see that the samples that were 30 minutes on the lab bench followed by snap freezing have much higher levels compared to the samples that were stabilized and then 30 minutes on the lab bench and frozen. So this is obviously the creation of this trimer is an ongoing process where it's being cleaved off from high molecular weight proteins in the upper area, and it ends up at this trimer. So an ongoing enzymatic process in the samples post-mortem or post-sample. That concludes the protein and the protein modification, and now I want to put some small molecule examples. First one we will look at using multi-imaging, looking at ATP AMP, and then the return to the fatty acids again, and another lipid, the shingle lipids example as well. We see here is an image, a MALDI image. It's an axial section of a mice brain, and it's been assayed then for ATP, the adenosine triphosphate. And we see in the heat-stabilized samples, we see areas of where we have local higher densities, and overall it's possible to get a signal. In the snap-frozen lower panel, oops, let me see that. In the lower panel, we have the snap-frozen sample. It's more of a sort of a general background noise that we see here. If we quantify these and make bar graphs of it, we see that in the snap-frozen sample, and then we also add AMP, the dephosphorylated version, so ATP converts to ADP and then to AMP. We see that the AMP levels are much higher in the snap-frozen sample compared to the ATP, which is what we would expect from a post-sampling condition where you have very low production of ATP. In the heat-stabilized sample, we have a much higher level of ATP compared to the AMP, and this is again what we would expect. If we don't have enzymatic activity, which is consuming the ATP, then ATP would be preserved to a much higher, Looking at fatty acids, this is the same image as earlier. I just want to highlight it again and to emphasize that we have an ongoing situation with an enzymatic activity in the sample releasing free fatty acids from the triglycerols primarily. And if we do the direct comparison, this is a direct comparison between the heat-stabilized HS and the snap-frozen SF, and it's done in brain and liver, and here I've highlighted 6 different of these free fatty acids. We see quite a remarkable difference in the levels between the heat-stabilized being, in all cases, lower or even much lower compared to the levels that we see in the snap-frozen sample. And the story is the same for brain as it is for liver. A similar story we see with sphingolipids or sphingosine. This is again a small interconnected system of molecules where we have sort of in the center we have the sphingosine, which can be phosphorylated into sphingosine 1-phosphate by adding this phosphor group here, and it can also be converted into sort of more of a lipid, a ceramide, by adding this sort of long chain down here. So what we see here, if we do a post-sampling time course, is that in the heat-stabilized samples, we have a small or a decrease over time compared to the snap-frozen sample, where you have a very rapid degradation in the post-sampling phase. So in this case, the samples are sort of harvested and then left at room temperature to decay. And we see here the phosphorylation is being lost very rapidly, and then it gets turned into the sphingosine. So we see that the sphingosine increase, and we see again the ceramide is also increasing in the snap-frozen samples, whereas in the heat-stabilized samples we see the levels are really retained. So once they're heat-stabilized, the levels are pretty much fixed. The lipids, these 2 publications are from 2 different publications that were released end of last year, beginning of this year, and we're now launching this, I think officially as of yesterday, as an application for heat stabilization. Analysis of nucleic acids in heat-stabilized samples. We we've seen that we don't preserve RNA. The RNases are very tough molecules. They do denature, but due to small size, and they're also held together, I think it's by 4 disulfide bridges, they tend to refold into a shape where into a functioning shape, so they can continue degrading RNA after. So it's not the examples where we see the phosphorylations, where heat-stabilized samples can be left at room temperature. That cannot be done with RNA. However, in some cases it's important to be able to do RNA-based analysis from heat-stabilized samples, and not so much that you want to preserve the RNA. And what we see here is that the RIN value, there is a bit of a drop, roughly 1 or so units in the RIN value, if we compare the snap frozen samples to the stabilized sample. It's, however, important to remember that the RIN value is primarily based on rRNA, and whereas most people are interested in the mRNA. And so there were some reports said that, oh yeah, heat will affect the rRNA, but the mRNA tends to be stable. So to investigate that, we worked together with a company called Tata, and they have a qPCR-based assay that will do mRNA qualification, quality assessment based on the mRNA directly. And they do this by first converting the mRNA into cDNA, and then they use 2 amplicons, so 2 primer pairs, to amplify first a short stretch and an overlapping long stretch. And the idea is that if you have degradation, you're likely to have more copies of the short compared to the long. So you will have a faster faster amplification if you have more copies of the shorter compared to a higher number of cycles needed to get amplification from the long amplicon if you have degradation. If you do the comparison between ordinary snap frozen samples and stabilized samples, and we do the comparison, so the left bar is the short amplicon, whereas the right in each pair are from the long amplicon. And we see here on the y-axis that this is the threshold of cycles where they start to into this exponential phase. And as we would expect, we see a very small difference, indicating that there is very little difference in the amplification between the short and the long. And if we look at the difference itself, we see here that although there is a difference between the long and the short, there is no difference in the samples that were heat stabilized compared to the snap frozen. And so the degradation assay and the stability assay, those are based on mRNA, whereas this is based on somewhere on the 18S rRNA. So to summarize the RNA story, I would say that RNA-based analysis is possible, but there is no stabilizing effect. So it's important to use some other type of combination effect, combination stabilization, if it's needed to stabilize the mRNA as well. So moving on to the last of the examples of what our users have been doing, and that's heat stabilization of pathogens, to inactivate pathogens. So this is an example. This has been developed by Lisa Casares. She works with the U.S. Army at Fort Detrick, and she wanted to do, generally she wanted to do multi-imaging on samples that were from a biosafety level 3 or 4 lab. And in order to get samples out from those labs, she needed to have them in formalin for roughly 21 days. And by then, the samples were so fixed, so it was impossible or near to impossible to do the mold imaging analysis that she wanted to do to identify viruses and distribution of chemicals and pharmaceuticals. So what she did, she wanted to use the stabilizer to quickly inactivate not only the enzymes but also bacteria and viruses in order to render these samples non-infective and to speed up the process of removal of these samples from these high containment labs out to the lab where she can have her mass spectrometer. So the sample, the experiment that she did to show that this was possible, she infected mice with a virus, a Venezuelan equine encephalitis virus, and there was also a bacterial pathogen, Burkholderia thailandiensis. These were two separate experiments. So one mice population was infected with the virus, one was infected with the bacteria. The animals were incubated to develop the disease, and then they were sacrificed, and she harvested various interesting organs there. And then she did these plaque assays, where she sort of basically homogenized it and then made a dilution series, and then plated it out onto plates to see where she had bacterial growth or where she had the virus, live viruses. And yeah, she did a comparison then between either just snap freezing the tissue or heat fixating it first using the stabilizer. And the results are quite conclusive. Using the standard for the bacterial case, we see to the left, the standard as what was previously called the snap frozen samples. She had very high levels of bacteria in the lung and the spleen, whereas the samples that were heat stabilized, these are small samples that are heat stabilized. So a small sample of some 3 millimeter size takes roughly 30 seconds to stabilize an instrument. There was no growth whatsoever. The virus is very similar. Here we really see that this is an encephalitis virus, so it's alive. It's in the brain. So in the snap frozen sample, in the brain sample, this is the dilution series. See there. Whereas in the spleen and the kidney, there is no activity because the virus is not replicating there. Whereas the heat inactivated samples, there is no activity. There is no virus at all. So these results will be published shortly, and I think this is an interesting application for some niches. This is another sort of general heat stabilization that we see in quite a few studies actually, an increase in reproducibility, so that the samples within a group cluster closer together are more similar. The left examples that we see here are this is a shotgun experiment, and we're comparing the identities in the intensities of the same identity in sample 1 and 2, and 1 and 3, and 2 and 3. And ideally, they would have the same intensity in all 3 samples. There's some that are higher intensity in one versus the other, and in some cases it's lower and so on. What's interesting though is that the spread in the heat stabilized group is much smaller compared to the snap frozen samples, where you really have outliers that are probably quite severely affected. So these peptides here, these phosphorylations here, are probably quite severely affected by the post-sampling process. Another way of visualizing this is from a completely different example, is to use a PCA plot, principal component analysis. So what we're seeing here is an experiment where we extracted low molecular weight peptides using various common buffers. So and then incubated it. So 2 molar urea, 8 molar urea, and so on. And what we're seeing here is that the D groups, that's the Denator Stabilized groups, regardless of extraction buffer, this is a 8 molar urea buffer in the PEP. The shotgun is extracted in 8 molar urea and then incubated for 6 hours as a 2 molar urea concentration. We see that they cluster very closely together there. If we look at the corresponding samples from the snap frozen samples, we see that the SF PEP and the SF shotgun, we see that they cluster, they're more spread apart within the group themselves, and also the groups are very different from one another. And our interpretation of this is that the changes that occur post-sampling and during homogenization and incubation in this case, they do have a tendency to change the samples in a certain direction, but there's also a spread within the group. And depending on the buffer, there are different sort of classes of enzymes active. So depending on the conditions, in some samples, you would have a tendency, as in this case, to move in one direction, whereas samples that were incubated in a different buffer, they had a tendency to go in this direction. They tend to hold together, although they also tend to spread out. So this is all published in a publication from last year. So where have our systems been sold to in the world? So we have roughly at the moment a little bit over 100 systems installed worldwide. So generally Europe, North America, and in Asia, Japan, China, North Korea, South Korea, and Australia. That's they tend to majority, I would say, is in academic labs, either doing sort of purely academic research or in connection with hospitals doing clinical research as well. We have a number of units with pharma companies, generally also for pharma companies with research divisions. There's also a number of instruments placed in biobanks, where they sort of act as a link between sample providers and people that do need samples. Yeah, so to finish off and summarize, unless it's been made obviously clear already, I think that that the analytes in the samples, they can change rapidly, and that it's very important to avoid this in order to keep the information that was in the in vivo sample as close as possible, all the way to analysis, so that you don't risk losing information during the analysis extraction analysis process. And to do this, of course, standardization of sampling, rapid stabilization, in order to stabilize the molecules of interest, and that the stabilizer system can be a vital part of a proper sample handling chain. That was generally the presentation that I wanted to present. As you realize, this is just a very brief snapshot of a lot of what we've done. There's lots more publications and information out there. So if you do have questions, as Mark said initially, we'll have a short Q&A session right now afterwards. And but it's also possible either to contact me directly at mats@denator.com. There's also some information on the webpage, www.denator.com. And so thanks a lot for listening. I hope you find some of it interesting and applicable to what you're doing.